Assalamualaikum and good morning, especially to Dr. Najis. Today, our group will present to you about the usage of the microbe, oil and gas. So, they have three group members of our group, consists of Nur Farhana Akila, Muhammad Hasif and myself, Muhammad Faiz bin Muhammad Azmi. So next we will go to the introduction. So before we go further, we need to know what is microbe. So when we talk about microbe, we always think about the disease that it can bring. Well, for a certain reason, they have a usage of microbes. Let's begin by defining some term which is microbe, clean up and oil. First, what do we mean by microbe? When we talk about microbe that are able to clean up oil, we are talking primarily about bacteria and fungi. Bacteria can break down oil to carbon dioxide and water. However, no single organism can break down all the components of crude oil or refined fuels spilled into the environment, and some bacteria can degrade several hydrocarbons or a class of hydrocarbons. Before this, we talk about the ability to clean up the oil, right? Especially when oil contamination at the sea. Now we will talk about the crude oil. So, crude oil are oil that are found in natural reservoir, which is principally derived from ancient algae and plant material. So, oil is a natural product. Therefore, the component of crude oil are great source of energy, not only as a fuel to power internal combustion engine. So, there will be have an activity of oil rig to get the energy source and slightly will be damaged by the contamination through the digging or through the transportation oil process. So, Halomonas species can isolate and characterize an efficient hydrocarbon degrading bacterium under hypersaline condition. This strange species is able to degrade hydrocarbon efficiently. The mode of hydrocarbon uptake is realized by the production of a biosurfactant which enhances the solubility of hydrocarbon and renders them more accessible for biodegradation. So we have the introduction of Halomonas species, and the Choosen microbe of our presentation is Halomonas species. So next we will want to know about the characteristic of Halomonas species. Halomonas species is a genus of halophilic, in other words is salt tolerating bacteria. Next, Halomonas species are gram negative rod shaped bacteria, and it generally have a sizing between 0.6 until 1.9 micrometer. It's very small. It also moves by using flagella, and it also can grow in the presence of oxygen, and have a reported latestly it can grow without oxygen. Fun fact about this Halomonas species, the species can display a pathogenic potential in human, so it's very dangerous to human species.
Next, I will present to you about the process in which Halomonas sp can be utilized in the oil and gas industry. First is bioremediation process. Halomonas bacteria have the ability to degrade hydrocarbons such as oil and gas contaminants. They can be employed in bioremediation process to clean up oil spills and contaminated sites. These bacteria break down the hydrocarbons into less harmful compounds, assisting in the environmental recovery of affected areas. Secondly, enhanced oil recovery (EOR). Halomonas species can be used in enhanced oil recovery techniques. Injecting these bacteria into oil reservoirs can improve oil recovery by enhancing the mobility of the oil. Halomonas produce biosurfactants which reduce the surface tension between oil and water, making it easier to displace and recover the oil. Next is souring control. Hydrogen sulfide (H2S) is a common issue in the oil and gas industry as it poses safety hazards and can cause corrosion in equipment. Halomonas bacteria can be used to control souring in oil reservoirs by selectively oxidizing sulfide ions and preventing the formation of hydrogen sulfide. This helps maintain a safe and corrosion-free working environment. Lastly is bioconversion of waste gases. Halomonas bacteria have the potential to convert waste gases such as carbon dioxide and methane into value-added products. This can be beneficial for the oil and gas industry as it provides an opportunity to reduce greenhouse gas emissions and generate valuable byproducts such as biofuels and bioplastics. Halomonas sp is a genus of halophilic bacteria that can be utilized for various applications in the oil and gas industry. This bacteria possesses unique mechanisms that enable them to thrive in high salinity environments, making them suitable for oil and gas related processes. Here are some mechanisms through which Halomonas sp can be utilized. Hydrocarbon degradation. Halomonas sp has the ability to degrade hydrocarbons such as crude oil and its derivatives. These bacteria produce specific enzymes such as alkane hydroxylase and dioxygenase, which can break down complex hydrocarbon molecules into simpler compounds. This degradation capability is beneficial for applications like bioremediation of oil spills and the treatment of produced water in oil extraction operations. Secondly, biosurfactant productions. Halomonas sp can provide biosurfactants which are surface-active compounds that can reduce interfacial tension between water and hydrocarbons. Biosurfactants enhance the accessibility of hydrocarbons to microbial degradation, increasing the efficiency of bioremediation process. They can also aid in enhanced oil recovery EOR techniques by facilitating the mobilization of trapped oil in reservoirs. Next, corrosion inhibition. In the oil and gas industry, corrosion of metal surface in a significant challenge. Halomonas sp can produce extracellular polymerase substances (EPS) that have corrosion inhibiting properties. These EPS can form a protective film on a metal surface, preventing or reducing the corrosion caused by corrosive substances present in the oil and gas production environment. Lastly, sulfur compound metabolisms. Halomonas sp has the ability to metabolize sulfur compounds including hydrogen sulfide H2S. H2S is a toxic gas produced during oil and gas production processes. Halomonas sp can oxidize H2S, converting it into less harmful compound like elemental sulfur or sulfate. This capability is valuable for mitigating the environmental and safety hazards associated with H2S. It is important to note that the specific mechanisms and application of Halomonas sp can vary depending on the strain and the target process. The utilization of Halomonas species for oil and gas industrial applications typically involves several stages. The first stage is strain isolation. This involves isolation and identification of Halomonas strains from natural environments. Secondly, strain screening. Evaluation of strains for tolerance to high salinity, temperature, and hydrocarbons as well as desired enzyme production. Thirdly, strain characterization. This involves genetic and physiological analysis of selected strains to understand their capabilities. Next is strain improvements. This involves techniques like genetic engineering or adaptive laboratory evolution to optimize strains for specific applications. Next is bioprocess development. This involves development of efficient cultivation and production methods for large-scale applications. Next is application testing. This is to test strains and their derived products in simulated or actual oil and gas industry conditions. Next is scale up and commercialization. To scaling up the bioprocess economics, feasibility studies, regulatory compliance, and intellectual property considerations. Lastly is industrial deployment. This involves collaborating with industry partners, providing technical support and continuous optimization for real-world conditions. These stages encompass the key steps involved in harnessing the potential of Halomonas species for oil and gas industrial applications.
Treatment in water, halomonase, which is in this case is alasan, breaking up the oil into tiny droplets and maintaining it as emulsion, improving its bioavailability to Halomonas sp that break down the oil. For instance, Microbes-Halomonas finista strain N32A is described as an excellently effective emulsion breaking bacteria. Emulsion breaking was low for the first 4 hours, but then it spiked in the 5th hour, then continued to rise steadily until it reached 92.5% after 24 hours. Treatment in soil. The addition of emulsifiers can increase the displacement of oil from soil particles, which facilitate the adhesion to oil droplets during emulsification. For example, Halomonas euryhalina H28 was isolated from saline soil and found able to produce the largest amount of exopolysaccharide substance, which is EPS, on glucose presence. The strains EPS capable of emulsifying crude oil at an efficiency of 80%. Treatment with nanoparticles. Combining nanoparticles with microorganisms that break down the oil considerably improve the percentage elimination of high oil concentration. The GSMS data from 4 Halomonas SP with the nanoparticles showed an oil removal of about 65 to 75% just after an hour. When both nanoparticles and bacteria are present, removal percentages dramatically increase and reach 100% after just 24 hours when compared to the initial oil-water mixture. Additional research is needed to determine the mechanism of oil removal. Future microbial enhanced oil recovery development plans include numerous workable strategies that shift the technology focus from microbial activity to the use of bioproducts.
So, for the current status part of this Halomonas species, I can say that it has been used and shown the positive result. I can give an example through article in 2009. They have a production water collected from Sersina Petroleum Reservoir located near Kerkenah Island, Tunisia. Halomonas species was used for the screening of halotolerant or halophilic bacteria and able to degrade crude oil. A halotolerant bacteria strain, Halomonas species, was isolated from production water of an oil field after enrichment on crude oil. The result was shown that Halomonas sp strain is able to degrade hydrocarbon efficiently. The mode of hydrocarbon uptake is realized by the production of a biosurfactant, which enhances the solubility of hydrocarbon and renders them more accessible for biodegradation. Other than that, they have a study in 2013 where the role of bacterial exopolysaccharide EPS in the fate of the oil release during the Deepwater Horizon oil spill. Halomonas species are recognized for producing exopolysaccharide exhibiting amphiphilic properties that allow this macromolecule to interface with hydrophobic substrate such as hydrocarbon. Halomonas species strain was shown to effectively increase the solubilization of aromatic hydrocarbon and enhance their biodegradation by an indigenous microbial community from oil contaminated surface water collected during the active phase of Deepwater Horizon oil spill. Then, Halomonas species isolated from the Deepwater Horizon spill site demonstrate its ability to effectively produce oil aggregate and emulsify the oil. The enrichment of exopolysaccharide producing bacteria during the spill coupled with their capacity to produce amphiphilic exopolysaccharide is likely to have contributed to the ultimate removal of the oil and the formation of oil, which were dominant feature observed in contaminated surface water. So, this microbe can be concluded have a big beneficial to be used in oil and rig industry and also have been proven by the latest article. So, that's all from us. Thank you.